So I'd like to take a look at post-translation modification, specifically phosphorylation of a protein, and what type of specific function that may serve inside of a cell. So what benefit does modifying proteins after they've been translated actually serve? Well, if you think about it, in a cell, this allows for not only optimization of function, efficiency, but also optimizing energy usage. If you look at my example here, if you start off with 3 proteins and just have 2 modifiers, do the calculation raised to the second power, you now have 9 different ways that 3 proteins can serve 9 different functions. You no longer have to make 9 proteins for 9 different functions. According to the Human Genome Project, we have approximately 21,000 genes. Now, this may not be a 1-to-1 ratio for the amount of proteins we have due to RNA splicing, editing, and such things. However, if you just take account for this number for what it is and apply even 7 or 8 chemical modifiers, now you can see how this exponential growth of different functions that these modifiers can serve. So here we can see a couple different chemical modifiers that can be applied to proteins. Some ones we know right off the bat: methylation. We know that when histones are methylated, it causes condensation of the chromatin structure, reducing transcription. We know that acetylation of histones relaxes chromatin, increasing transcription. But again, I want to take a specific look at phosphorylation, adding a phosphate group to a protein. We know this is done through kinases, and we know that the removal of phosphate is done through phosphatases. In this diagram here, there was a published paper in Nature that took an analytical look at the SwissProt database, which is a database of sequenced proteins. And they wanted to take a look at the approximately 530,000 sequenced proteins and establish a rate for post-translational modifications. And what they found is that phosphorylation had the highest rate. So at face value, I believe you could ask the question as to why the cell is choosing phosphorylation as a mechanism over these others. So I'd like to add a little background information. Back in 1906, by an experiment by Levin and Alsberg, identified the first phosphate group existed inside of a protein. The protein that they looked at was vitelline. In the 1950s, an experiment done by Fisher and Krebs, what they found is that the enzyme glycogen phosphorylase, which catalyzes the reaction of inorganic phosphate plus glycogen to glucose 1-phosphate, was activated by the addition of a phosphate group by phosphorylase kinase and was inactivated by the removal of the phosphate group by phosphorylase phosphatase. This was huge. It established a way for the cell to have a regulatory mechanism to turn on and off enzymes, basically establishing an on and off switch. This was huge finding. Before, the thinking was that if you wanted a protein, you had to make it and then degrade it. And we know that that didn't coincide with Mother Nature, because we know she likes to pinch pennies and save energy when she can. This was huge again. Just to show you, they won the Nobel Prize in 1992 for it. So here in this diagram, we can see that phosphorylating protein serves many functions, from metabolism, transcription, differentiation, to transport. But I want to take a specific look at cell death, also known as apoptosis. In this published paper, they wanted to take a look at the PAR4 gene, a gene known to encode for a tumor suppressor protein that induces apoptosis specifically in cancer cells, but not in normal cells. PAR4 is known to be diminished or deleted in many forms of cancer. What they knew about PAR4 is that it needed to be mobilized into the nucleus of the cell for it to induce the caspase pathway for activation of apoptosis in the cancer cell. They set out to establish and find a mechanism as to the specificity of apoptosis only in cancer cells. And what they found is that when PAR4 is phosphorylated, at its threonine amino acid at the 155th position. This allows for it to mobilize into the nucleus and have a specific induction for apoptosis only in cancer cells. This was a novel mechanism that they found. And what they also found additionally is that protein kinases, which phosphorylated the T155th residue, is elevated in transformed cells and cancer cell lines. In this top figure, we can see that we have PAR4 gene has a high percentage of apoptosis. When you point mutate PAR4 at the 154th position to an alanine, you still have a high rate of apoptosis but with a slight decrease. However, when you point mutate the threonine at the 155th position to an alanine, you have a severe decrease in apoptosis. In this bottom figure, they wanted to show that PAR4 was being phosphorylated at the 155th position. They used P32, and as you can see, in regular PAR4, you have phosphorylation. When you point mutate the 154th position, you still have phosphorylation with a slight decrease. However, when you completely change the threonine to an alanine in the 155th, you have a severe drop in phosphorylation. In this top figure, we have 3 known cancer cell lines, and we have an immortal and normal cell line. As you can see, when they measured the pKa activity, you had a high rate of pKa activity in the cancer cell lines. In this bottom figure, they used green fluorescent protein, and what they did is that they labeled it with PAR4, and they used pKa, which is a protein kinase inhibitor, to show that when you inhibit the ability for PAR4 to be phosphorylated, you have this decrease in percent of apoptosis. This diagram provides a summary of the published paper. As you can see, when PAR4 is phosphorylated at the 155th position at its threonine residue, it allows PAR4 to be mobilized into the nucleus and induce an apoptosis pathway. When you have no or low pKa and you cannot phosphorylate PAR4 at the 155th position, you cannot mobilize PAR4 into the nucleus, and you cannot induce an apoptic pathway. So what did this establish? It established that protein kinases, which mediate the phosphorylation of the T155th residue of PAR4, was essential and a selective mechanism for the function of PAR4 as inducing apoptosis specifically in cancer cells. Additionally, they found that the enzymatic activity of pKa was elevated in transforming cancer cell lines, and this establishes another selective mechanism for activating the specific function of apoptosis of PAR4 in cancer cell lines. Thanks for watching. Make sure you subscribe and give me a big thumbs up.